Good morning or good afternoon, everyone. Welcome to the Xylella Seminars. I'm Luis, a postdoctoral researcher from the Institute of Sustainable Agriculture, and I will be presenting today's speaker, Aniol Boisac. Aniol graduated with a degree in food innovation and security in 2020 and earned his master's degree in food biotechnology in 2021 from the University of Girona. And in 2020, he began to work with the feed subgroup on projects focused on quarantine pathogens, including Xylella fastidiosa, Candidatus leberibacter, and Nervinia milogora. His role involved participating in public research projects at both the Spanish and European levels, as well as facilitating technology transfer through agreements and collaborations with various companies. In 2022, he started his PhD, focused on control methods for Xylella fastidiosa, supported by a predoctoral contract from the Bexil project. The work that he's presented today is titled Aggressiveness of Subspecies and STs of Xylella fastidiosa in the surrogated host Nicotiana benthamiana. To the audience, please remember that if you have any questions, feel free to type them in the chat, or at the end of the presentation, you can ask them directly. So, Aniol, whenever you want, the floor is yours.
Okay, thank you, Luis, for the introduction. Well, welcome, everyone. I'm Aniol, and as Luis said, I will present this work, which is a part of my PhD thesis. First, a brief introduction about Xylella fastidiosa. As you probably know, Xylella fastidiosa is a gram-negative bacteria characterized by its xylem-limited colonization, affecting more than 700 plant species, and that its transmission is done by xylem-feeding insects, where the primary vector in Europe is Philaenus spumarius. Some important hosts affected by Xylella fastidiosa are grapevines, olive, almond, and citrus. Its first detection in Europe was in Italy in 2013, and it has already established in France and Spain. It's classified as a quarantine pathogen, and there aren't any effective treatments for the control of infected hosts. Another particularity of Xylella fastidiosa is its genetic diversity, where we can find 5 main subspecies, and each one exhibits distinct host range. And also we can group the different strains according to their sequence type, where there are also differences in host preference between them, as we can see in this graph for the pauca, for the subspecies pauca. This range of hosts makes the comparison between strains difficult because of the lack of a framework in which all the different strains can infect, colonize, and develop symptoms in the same conditions, including the same host. One of the objectives of this work was to establish this framework, and just so you know, all the results that I'm going to show you were published last month in this article, which is still in first look, but you can access it already. Okay, so we have 4 principal objectives. The first one, to validate Nicotiana benthamiana as a surrogate host for Xylella fastidiosa studies, to characterize the aggressiveness of several Xylella fastidiosa strains from different subspecies and sequence type, to determine if there is a relationship between aggressiveness properties and subspecies, and to relate symptoms development and aggressiveness with the host defense response. Globally, we want to provide available insights for epidemiological risk assessment and the development of targeted disease management strategies. Okay, so to assess the aggressiveness profiling, we use Nicotiana benthamiana as a surrogate host, and we use the precision microinjection method to infect the plants with a suspension of 30 microliters of 10 to the 8 CFU per milliliter suspension of Xylella fastidiosa for the 17 strains that we use. We took samples at 5 different times, and we used 3 plants each time. We sampled 8 centimeters of steam above the inoculation point and then we did the DNA extraction using a commercial kit and performed the qPCR analysis to quantify the Xylella fastidiosa population in the plant. We also evaluate the symptoms according to a severity scale that goes from 0 to 5. Don't worry if the image is too small, because I will show you more photos of symptoms later. And before moving to the experimental design for the transcriptomic analysis of the plant, I would like to show you the list of the strains that we use and also the standard curves of the strains. Here we have the list of the 17 strains that we used. The subspecies, the sequence type, and also the origin of the strains, and the standard curves of the strains in Nicotiana benthamiana extracts. I would like to take also a moment to tell you the importance of performing the standard curves of the strains to be able to compare aggressiveness parameters between them. Because as you can see, if we took a CT value of 20, for example, okay, let me see if I can. Yes. Okay. So, for example, now if we took, if we have a value, a CT value of 20, for example, for the Pauca strain, Chill 1961, we talk about 6 log CFU per milliliter, but the same CT value for the fastidiosa strain EVIA 5388, it's almost it's between 7 and 8 CFU logs, the CFU per gram. So if we were only work with CT values, we will say that these two strains are similar in colonization capacity, which is not real. And also doing these curves, it's important because we can see if there are any qPCI inhibitors in the plant extract, and also it gives us the limit of the technique. Okay, so for the transcriptomic analysis, we also use the Nicotiana benthamiana, and we perform the same inoculation method. But in this case, we only use the 3 strains from the subspecies Pauca and sequence type 53. I will tell you why in a moment. And for the analysis, we work with a group of 9 plants, and we took half a leaf for each plant that were grouped into 3 randomized biological replicates. After that, we did the RNA extraction using the PureLink reagent and performed an RT-qPCR analysis of the 19 selected genes. And finally, we used the delta-delta CT method to quantify the relative gene expression. We sample at 3 different times, 4, 15, and 30 days after the inoculation. Okay, so moving to the aggressiveness results in the graph. Okay, sorry, because it's a heavy slide and sometimes take times loading. Okay, so in the graph, you can see the colonization dynamic and the symptoms development from 7 to 42 days after the inoculation, and the strains are grouped in subspecies and sequence type. First thing that we can see is that there is an effective colonization of all 17 strains, and we also identify two different trends. We found that what we can call the high colonizers, like the fastidiosa and 1 multiplex strain, that reach between 6 and 7 logs CFU per gram, and the low colonizers, where we can find some of the Pauca and multiplex strains that reach only 4 logs CFU per gram. So we already see some difference in the colonization capacity between strains of the same subspecies and sequence type. Also, all strains cause some sort of symptoms in the plants, confirming the susceptibility of Nicotiana benthamiana to Xylella infection. And there is a pattern in the disease progression where after the apparition of the first symptoms, uh, there is a period of with no progression and followed by an increase of the symptom severity at the end of the experiment. In order to find if there was some relationship between cell concentration and symptoms development, or some relationship between strains and sequence type, we work all these data into the area under the growth curve and the area under the disease progression curve to integrate all together. But before seeing that, I will show you some photos of the Nicotiana benthamiana symptoms. Just a moment, because this is a heavy slide and it takes some time also because there are a lot of photos. Okay. So in these slides, we can see some Nicotiana benthamiana plants infected with different Xylella fastidiosa strains and at different times. So we can see some of the typical symptoms. Symptoms are strain dependent, and they go from a general decay that can affect all the plant, or just here you can see that half of the plant presents some decay and the other half is okay. Also, we see some necrotic lesions that can be more general or like spots. This is a photo of necrotic. I don't know if you can see quite well because it's not chlorosis, it's necrotic lesions. It's like transparent, it's not chlorotic, but we see some chlorotic in other strains. There are chlorosis. And also here we can see some dead plants or almost dead plants. And some strains cause a mix of different also symptoms, like some decay, some chlorosis, necrotic lesions. So we have a range of different symptoms. And if I would like to give you some tips from our experience working with Nicotiana benthamiana, that is always have a control group of non-treated plants, non-infected plants, because, for example, in our case, we use the microinjection method. So our control group is microinjected with the media, the PD2 media. So you see if during the experiment there are some problems, or you can see if plants has some kind of a stress. Also, if it's possible, having a control group of infected plants with a strain that you already know how it performs. Like you know how much time you need for the prevention of the first symptoms, the typical symptoms, also the population levels that you expect if you are performing qPCR, especially if you work with new strains, because as you can see, symptoms are can be different depending on the strain that we use. Okay, so as I was saying before, we work all the disease and population data into the area under the curve formula to see if we could establish some kind of relationship. And as you can see, there isn't a relationship neither between population growth and disease severity, nor between species or sequence type. What we found were two main groups of strains that present similar characteristics. A group of strains with similar cell concentration but a wide range of disease severity, and another group with different cell concentration but similar severity levels. So virulence is driven by more than cell concentration and vessel occlusion, as can be quorum sensing regulation, toxin and enzyme production, extracellular polysaccharides, and the host immune modulation to the infection. And to explore this, we did a transcriptomic analysis of the Nicotiana benthamiana plants with the subspecies Pauca sequence type 53. Because, as you can see, they are the ones with the most similarities between them in terms of population severity, of population levels and severity disease, and also for the implication of the Dedona strain in the Italy outbreak and the implication in the olive production here in Spain. Before going to the transcriptomic results, I also want to present the dose response assay that we performed with these strains that are also in the article. Okay, so here what we did was inoculate different groups of Nicotiana benthamiana plants with an inoculum of Xylella fastidiosa going from 300 CFU per plant to 10 to the 7 CFU per plant. And we evaluate the symptom severity and the population levels after 40 days. Here we have also the fastidiosa strain EVIA 5387. This is our reference strain that I told you when we saw the symptoms. We did a lot of work with these strains, and we know how it performs on Nicotiana benthamiana, what to expect, in terms of population levels and symptoms development. So here, all the strains present the typical S-shaped saturation pattern, and interesting, we observe symptoms in all inoculated doses. So we established the minimum effective dose to add 300 CFU per plant. That was the lowest dose that we use. This is important and can have some implication for risk management because, for example, the detection limit by qPCR of Xylella fastidiosa in Nicotiana benthamiana is between 100 and 1000 CFU per gram, depending on the strain. So we can have symptomatic plants infected by Xylella, but that we aren't able to confirm by qPCR analysis. So this opens the door that maybe it will happen also into other hosts. Okay, so finally, the last results are the host transcriptomic response to the infection of the tree pauca strains. In the graph, we can see the expression of the 19 selected genes for the three strains and at the different times during the infection. Genes are grouped into the salicylic acid pathway, jasmine acid and ethylene pathway, transcriptomic factors, metabolism-related genes, and programmed cell death and growth response-related genes. Here we observe two different things. First, that there is a strain-dependent response modulation of the plant, and also that Xylella fastidiosa can avoid the defense response of the host. Because, as you can see, at early stage of the infection, there is an initial recognition of the pathogen and an activation of the salicylic acid pathway. But as the infection advances, we observe these two things, that there is a different response for each strain of the plant, and also a downregulation of the defense-related genes that it's greater at 30 days after the infection. Well, here there is a strain comparison between the CN28 strain, which has the most number of upregulated genes, and the Dedona strain which has the lowest number of upregulated genes, which is also the one that had greater population concentration, if you remember on the first graph, but was not the one that caused most symptoms, if you remember. It was the GP 18 strains. Here, well, there is a recap of all the results that from the different assays that can be synthesized in the phrase, no, strain-dependent pathogenesis and immune evasion. And finally, the conclusions of these different experiments was that we confirmed the suitability of Nicotiana benthamiana as a model plant to conduct Xylella fastidiosa assays, that there is a wide diversity of aggressiveness observed across the 17 strains. No direct relationship was found between population growth and symptom severity, that there is a significant variability in aggressiveness parameters even within strains of the same sequence type, that there is a strain-dependent modulation of the host transcriptomic profile, that globally the transcriptomic profile is characterized by an early recognition of the pathogen followed by an immune evasion at later stage. And this has some management implications. For example, to incorporate a strain-specific aggressiveness data into epidemiological risk simulations, to know that the low minimum infective dose could be a critical risk for disease spread, and that control strategies must account for the host inability to maintain a long-term immune response. And of course, since these results comes from a controlled framework experiment using a surrogate host, aggressiveness should be evaluated in an extensive range of hosts and conditions in order to make extrapolations to woody trees as olive or citrus or almond. So thank you for your attention. Just to remember, these results and more detailed data are available in the article. And if there is any questions, I will be glad to answer them.
Thank you very much for your presentation, Aniel. So, as Aniel mentioned, if you have any questions regarding his work, please feel free to write questions in the chat or open the mic and ask them directly. I